welcome to DNA replication, the instructional video. Here I'm going to animate using PowerPoint a DNA molecule being replicated and the process and enzymes that are involved. This is a simple DNA strand and it's helicase, and he comes in and unzips these genes, and what he's doing is he's breaking the hydrogen bonds that are between these bases here that are labeled with letters. We'll learn what those mean in a minute. RNA primase then adds the RNA primers to each strand. Now what these primers do is they give polymerase, it's kind of like a flag to tell it where to start and what to pick. Polymerase picks these up and then places down the appropriate bases. This only occurs in the 5' to 3' direction. DNA polymerase then moves in the 3' direction, adding bases to the template strand. And this is the template strand here, and the appropriate complementary base has been added. So what are the 3' and 5' directions? Carbon and a molecule of a nucleotide. We can see going clockwise, starting at the oxygen. So the oxygen right there at the top. So it goes 1, 2, 3, 4, 5, and the 5' end has the phosphate on it, and the 3' end is what connects to the phosphate of the prior nucleotide and a chain. So here you can see where they are bonded, and the 5' end always ends in that phosphate group, and the 3' end always ends in the third carbon. So polymerase proceeds in the 3' direction. So that's the same with helicase, and that's the same with helicase, and that's the same with primase. So polymerase on the other strand, however, so we have two different strands here. It has to enter from the 5' to the 3' end, and because these strands are anti-parallel, that creates kind of a problem. In one strand we have the entire shoelace to get, and on the other one we're only getting what we feed into the hole. The strand where the 5' end continues, it seems to grow, is called the lagging strand. So because helicase travels in the 3' direction, the lagging strand has to be replicated in small portions. These small portions are called Okazaki fragments. And then ligase comes in, and this ligase acts as the glue that brings it all together. So what it does is it makes those phosphodiester bonds. The phosphodiester bonds are the 5' to 3' ends connecting. So it's right between that phosphate and that carbon right there. Ligase makes these bonds, and that's what makes that backbone. And these bonds are covalent bonds. So they are very strong. And then the end result of replication, of course, is two identical DNA strands. So as you can see, each one is containing the one strand of the original DNA molecule. And one is containing the leading strand, and one is containing the lagging strand. Thank you for watching my video. Hope you learned something. Follow the link below to move on to the assessment portion. Thank you.